annotations in some format, and then you did your reflection, and that'll be good for me. And the idea behind that was just so that you know for the other labs, like how in detail should you be. And then the final assessment of that is done by me, and I do it at the very end. So after all your labs are done, so you have plenty of time like to go back and fix and fill in and things like that. So for today, I wanted to, since I did give you the translation as a video, I want to make sure there was no confusion about that. So we'll talk briefly about that, and then we'll get right into the gene regulation and prokaryotes. So hopefully we'll cover that in a single day. For lab this week, it's exciting because we're going to actually get back to the worms, and we're also going to be checking our sequences. So I quickly looked at some of the sequences. The two that I opened looked good. So today you'll have a chance to actually check to see if the sequence that you got is what you expect. And then I started for everybody this weekend. Each group has one plate with GMC 101 and one plate with N2. So today I'm going to actually show you kind of how to do staging of the worms, or how to figure out the correct age. Because as you'll see, for the paralysis assay, we're supposed to pick L4s, which is a certain larval stage, and put those onto a new plate, and we'll place them at the 25-degree incubator. And I'm hoping there's no problems because there's a power outage scheduled for tonight in this building. So anyway, hopefully things go well. But in the meantime, you'll have a little practice to look at the worms and try to find the different larval stages. You can practice picking today if you want, but tomorrow will be the real big important day where we're actually going to pick and put them onto new plates. Okay, so that's sort of the plan. The paralysis assay will take all week. So tomorrow we'll be putting them on, putting the L4 worms onto the plates and placing them at 25 degrees. And then on Wednesday, we'll be scoring day 1, which means that you're going to do this touch assay. So did anybody have a chance to kind of read about it, or do you remember how they do the touch assay to test for paralysis? Yeah, with your little pick, you just touch the nose. The idea is that if they're not paralyzed, they should actually retract and go backwards. If they're paralyzed, they can, initially when they first are paralyzed, they can still move their head up and down, but they won't be able to back up. And when it's full paralysis, they don't move at all. And I'll kind of talk a little bit about how can you tell if they're dead or they're paralyzed. You might have to kind of classify that as well. So we'll do scoring 24 hours after they've been put down, so that's, or put on the plates. So that'll be Wednesday. And then Thursday we have our second exam. But after that, you'll do the second scoring. So the same thing, just touching them all and keeping track. So for that, I did want to mention this before we get started. For your pre-lab for lab 7, the best reference is to go back. Do you have that paper? Yeah, go back to this paper that we read for the journal club. The one that was called Utility of an Improved Model. In the method section, they describe a paralysis assay.
On page 7.
Okay, yeah, so you must have been looking at it. There is a section where they do talk about how they set up.
It's like the smallest paragraph.
Okay,
Page 8.
Oh, sorry, 8.
page 8. Oh yeah, it's down here. So paralysis assay starts here. They talk about how they did this. The other thing to look at is numbers, because with science, right, it's really important to figure out how many should I put in order to be confident with my results, right? So for that, you're going to consult with the person that you're working with, and just keep in mind that you have to be able to pick those worms and move them. So we're going to kind of collect all of our data, and we'll report it as a class, so you'll be able to compare your data set to the classes. We're all working with the same strain, and that way we can increase the number of worms, the N number that we have. But just try to think with you and your partner, like, what is a reasonable number for each strain? We have N2 and GMC 101, and just keep in mind you're going to have to be able to move them. I will help. I know it's like the second time you're picking worms, so if you really struggle and you just can't get any worms, I will help to pick the worms. But think about what you want to do. Yeah.
And this in this assay, are we going to be feeding our plasmids to the worms as well and checking for cows?
Eventually, yeah, so this first round is no RNAi at all. It's the Senior Abditis Genetics Center. We just want to confirm that we get similar results, right? Because we're using these incubators, but there can be all these small variations, and so we're gonna compare our numbers to the numbers they have in the paper.
Got it.
As a first round. And then the second round, if you have confirmed your RNAi, then you'll get a chance to do the RNAi and then do the paralysis assay.
Thank you.
Okay, so you'll only need a pre-lab for, obviously today you needed one and then tomorrow, but after that, it's all part of lab 7 for this week. So, anybody have any questions? I was also thinking Wednesday, we can do a review session on Wednesday before the exam on Thursday as well. So this stuff that we're supposed to be starting is actually not on the test, so we can push it back if we need to. Okay, so then, oh yeah, and I think Crystal handed out the ICA for today. So today we are gonna start with the gene regulation. Before we do that though, I did want to do a little bit of review about translation. So I think most of you had handed in the ICA for translation. But let's talk a little bit about the differences between translation for prokaryotes and eukaryotes. And that would be actually we can draw a comparison up on the board. So if you want to open up your notes on translation for a second, there were some differences between translation in prokaryotes and eukaryotes. And on the ICA, actually, see if I still have a copy of it here. So in this picture of the ribosome here, this was for a bacterial ribosome, right? So they have some features on here. And then I think one of the questions, question F says, how would it be different if this was a eukaryotic ribosome? So what did people think? Yeah.
Well, asthma is bacterial, right?
Yeah, so this one, we have translation. And then for prokaryotes, they use formal methionine as the first amino acid. And what do eukaryotes use? Yeah, so that's one difference. What's another difference? Yeah.
Prokaryotes try to look for the shine down with the L-Garno sequence versus eukaryotes they use the COSAR.
Yep. Okay, so to position the ribosome, they use the shine L-Garno sequence, and remember that was actually a part of the ribosome itself. So I'm going to draw this here. This was the small subunit, and then there's this little piece of RNA. Does anyone remember what the RNA itself was called?
16S.
Yeah. So there's the 16S rRNA that pairs up with the shine L-Garno sequence. That's part of the mRNA. So this was how, in the beginning of translation, bacteria could figure out where to place the very first AUG. So if it positions the shine L-Garno with base paired with the rRNA, then the AUG will be right in the P site, right where it needs to be. So this would be the P site. And then you mentioned COSAC sequence. So for eukaryotes, they use the COSAC sequence. And you don't really have to know the exact sequence, but basically it's sort of a context where the AUG is found with certain bases close by. And instead of the ribosome being the thing that can feel for that, anybody remember what piece of the machinery can tell where that COSAC sequence is? So it's not actually the ribosome, but instead it's another RNA. It's the tRNA.
Oh,
So it's the tRNA, the initiator tRNA that comes in. It has additional contacts that can sort of feel around. Sorry, this pen is sort of slowly dying. Yeah. So there's the tRNA, which has the methionine on it, can kind of feel that, okay, this AUG has this context, this Kozak sequence. All right, any others? Any other things that were different between prokaryotes and eukaryotes? Yeah, Daphne?
Is it like eukaryotes have a circle of translation?
Yes, yes. This is really important. So they use the circle, quote, circle of translation. Can anybody describe what that means? Circle of translation, like the circle of life.
so the mRNA gets a cap on the 5' end and a poly
Yeah.
tail on the 3' end, and then they make a circle.
Yeah. So you have your 5' cap, and then that goes around in a circle, and the 3' poly A tail is here, and the proteins that the initiator proteins bind to both. And can anybody explain why would you want to do that? What's the purpose of bringing them together to form like a circle?
So now they're ready.
Yeah. So if you were, let's say that you had an RNA floating around in the cell that was chewed up, like, and missing, it had been half chewed, it would be completely wasteful to start translating that because you can't get to the end of it. So this is a quality control. So it's a quality control check. All right. But in prokaryotes, do they do that, or is there any kind of a regulation on when it can start? It can actually start as soon as you have transcription and you have a little bit of RNA, you can start translating it, right? So we said that in prokaryotes, they can actually do that co-transcriptional translation, which means that basically you can do it all at the same time in the same place for efficiency. But obviously the benefit of this is kind of higher quality and not wasting energy. Okay, and then the only other concept I wanted to kind of go over is the idea about the wobble. So can anybody from the video explain what wobble means?
There's only so many tRNAs, and there's like 60 possible combinations, and so the last letter is variable.
Yeah, exactly. So it's like permissive. So I gave the example in the video if you go like this with your fingers. This one is farther apart, so those contacts, that's actually kind of what's happening as the RNA is bending, is that there's only tight base pairing in the first two. So for example, if you had CGU in the mRNA, if this is 5' to 3', this one can have other base pairs other than just A. And you don't have to know all the details because there are very specific details about what can base pair with what. But the idea behind wobble is that the tRNA that comes in, the anticodon, is going to be G, C, and then this position could be variable. And so that is referred to as wobble. The only other note I want to make, because this is a common misconception and it causes a problem on the exam, is when you're going from an mRNA to a tRNA sequence, we'll put the A in there. One of the most common problems is that people forget that if this is base pairing with another nucleic acid, what happens to the directionality of those? So this would be our codon, and the anticodon would be GCA, but what do we have to show with the directionality? It has to be antiparallel. So just make sure in your mind, because I think I have a question that actually gives you, we could do this for practice right now, but if you had, we'll say that the coding DNA strand So now we're not even in RNA, but if you had the coding DNA was can't be U, ATC, then what would the anticodon, and I'll put the sequence 5'3'. So with the person next to you, see if you can get from this coding DNA all the way to the anticodon, and with the right orientation.
Changing the yeah, but then you said
AUC?
no?
Yeah.
Consent, well,
Yeah.
It's AUC.
Oh, the A would flip, wouldn't it?
AUC, and then you would look on here, the
So it'd be
anticodon
G, U, C.
would be,
Yeah, CAG 3 to 5.
Mhm.
G, back to 5, because they're all anti-parallel.
Mhm.
So back and forth.
UAG. So if you're UAG, and then that's what you're looking at, and then UAG, that one you have. Unless you flip it and it's G and U.
Or no, so the AUC would be,
The AUC, that's the
I
empire.
had never heard that done.
yeah,
Mhm.
but then
But I don't really know. So I did like
Can I ask about it? Which is a bit different, basically. I just wrote down that we're going to analyze the DNA sequencing.
yeah. So they're comparing the other one to the other one, but then the only comparison that the compare steps that I found is a protein or something, and I could find the steps where comparing to the same kind of.
Okay.
So I don't know if it's gonna be the same steps, but it's like so I had like two. I know that's true.
Sounds probably what I heard too. This is very vague.
I know it's really. I had like two steps because I was afraid that the first step was gonna be wrong, so I wrote like another one. I was like, or this one could be wrong.
I'm confusing myself with the ICO.
And then completely wrong, so
Oh, there it is.
oh wow.
Open, analyze, copy from well. Okay.
Literally, like I can't. I don't even know what I wrote.
Okay, so for this, if this was the coding strand, let's just kind of do it in the order that you did it in your head. So if you're at the coding strand, what's the next easiest thing to figure out? Yeah, the mRNA. So what would the mRNA be if that was the coding strand? So we'll keep the same orientation, AUC, because remember this is the non-template, so they do have the same orientation. And then we go from the codon to the anticodon, or the tRNA. And what would that one be? Yeah, U, A, G, and then we have to flop these. So this would be 3 prime, this would be 5 prime. And then what would that code for? LIE? Or ILE?
Oh, that's an I. Then yes,
Yeah,
ILE.
okay. Right, so isoleucine is that one, so ILE. Did everybody get that one? Okay, so I know that that's kind of one of the ways that I ask questions. You have to be able to go from DNA to RNA to tRNA, and sometimes I don't ask about the middle part, just say like, if this was this part, what would be the anticodon? So that's a good thing to kind of practice going back and forth between DNA and RNA and the tRNA. Okay, anybody have questions from any other parts of the translation
Just
lecture?
clarification.
Yeah.
Because we were discussing it, and I want to make sure that it wasn't just helping our lives.
Yeah.
So it goes from coding DNA, and then it goes to mRNA, and the protein, little blocky guy over here, is read from, that's where we get the ILE from, right? And
From
then it's
mRNA.
Yeah,
Yes,
And
this,
then it's
you use the chart with.
yeah, and then tRNA is a little T that comes in with the little ILE attached to it, and it goes, oh yeah, you have ABC, and then here, take my ILE, bye.
Yes.
Right,
That
okay.
was right. Yeah, and actually, I think Matt had a question, yeah.
Well, I was just wondering, so DNA is always written 5 to 3, and then RNA isn't technically
So,
necessary.
DNA, we have 2 strands of DNA. Conventionally, if you're just given a sequence, usually you assume it's 5' to 3', and like when gene sequences are given, usually the coding strand is given. But, because there's 2 coding and template, if I gave a question like this, I would always indicate like what 5' to 3' is.
Just for clarification, the tRNA synthetases, they're in both eukaryotes and prokaryotes, and they're in the decoding center? Is that?
Yeah, they have so the tRNA synthetase is floating around the cell separate from the ribosome, and it's like a charging station. Like when you have an empty tRNA, it charges it up with the appropriate amino acid. So it does have to feel, that's a good question. The tRNA has this kind of, it's like a cloverleaf structure, and this is attached, I actually forget, I think it's attached to a 3' end, and there's always this characteristic CCA at that end, but there's 2 parts that the synthetase, so the synthetase is like a big enzyme that's gonna bind to this and to the tRNA, and it's kind of checking to make sure that this sequence that's in the anticodon loop is the appropriate sequence for that amino acid. So it does, that's where all the checking occurs, to say like, yes, this tRNA has the right amino acid. After that, the ribosome is never gonna check. So whatever's floating around the cell, if it's charged with the wrong thing, it'll just put the wrong amino acid into that protein.
Oh, okay, got it. Makes sense.
Yeah, so the ribosome, I think there's also a question about that, or like a concept check that says, does the ribosome ever check? And the answer is no, just whatever's charged up, that's what gets inserted in. Okay. All right, so I think you have pretty good understanding for translation. So, do people care if I record? I was gonna record, you'll only probably be able to hear my voice because I always walk over here, but then the camera's like facing off over here. But that way it'd be on Panopto. And then, but I just wanna make sure you guys are okay with it because if you talk, then your voice is recorded, so. I think I can, you know what, I actually never even put Panopto in our class stuff because I thought, well, we're not doing recordings, but I'll just drag that up here.
Will the camera be able to see the whiteboard and what you write on?
So I can angle it, that. But it will record whatever's like, whatever PowerPoint is pulled up, I think, so that's typically how it works. Okay, let's just see, we'll give it a test run today. And then for future reference, if any of you want to find the recordings, you'll just go to the Panopto recordings button. And you should be able to find all of the ones that have been saved for our class. Oh yes, I have the good old days of, oh no. So can I ask you all, how do you like having class 100% in person versus, I mean, you've been doing online school for a while. What are the pluses and minuses of it?
I miss being able to multitask because like I mentioned before, like I have 2 laptops usually in front of me and then 2 screens above those, and so I have really great setup. So for lectures, it's really nice, so I can listen to the lecture and work at the same time.
Yeah.
But,
How about others? Yeah, Matt.
Well, I like that when lectures were recorded, I could go back and pause and then draw stuff out or take notes and not have to rush
Yeah.
on to the next one. Really.
Yeah. I
Doing stuff.
I do like in person. So
Yeah,
I feel like in person there's less friction to ask a question, and anytime I was online, you'd have to like unmute yourself, and then always my brain would go, nah, you can get it later. You don't have to ask
Yeah,
now.
that's kind of intimidating, yeah, to unmute yourself.
Yeah, I would say being able to ask questions in person and having like to see drawn out on a board and like the engagement is better. But I also think that not having to do 3 hours of mastering chemistry or mastering biology homework or whatever is really nice. 'Cause
Oh, yeah, I
that
just
could
do
go
that.
over.
Yeah,
So
yeah, how about
I really enjoy the in-person labs.
the lab?
Oh, yeah.
Yeah,
A lot
that's
nicer.
true. There's really no good substitute for that.
No.
So guys, I don't have space. Unfortunately, I'm one of those people that never cleans up stuff. We'll do this tomorrow though. So, if anybody wants to record like audio, I don't care. So you can do that if you want. But in tomorrow we'll get this fixed up. And now I just need to be able to put this up. Okay. Yeah, it's kind of interesting because our whole department is, talking about how should we continue to offer classes and what classes people will sign up for.
I do feel like driving here 4 days a week is like so much anxiety and stress. Like I literally will like be driving here and be like, just kidding, take me home. Like
Yeah, that's true.
So
Okay,
gas
that's nice.
is
Yeah, the gas.
Yeah.
I like coming here for labs, as doing the online school, I really missed like the hands-on part of labs.
Mm-hmm.
I think the balance is like a hybrid, but so yeah, for me personally, delivering, it's very like sad and depressing when nobody turns their camera on, so you're just like talking to like a wall of black screen. So, and then also like if you tell a joke and nobody's so anyway, that part I like, but okay. So today we're gonna do this first part 3. We'll go through prokaryotes. It's actually about 37 slides. I think we can get through it today. Then tomorrow we'll start the regulation of gene expression, which takes a little bit more time, and talk about like chromatin and nuclear architecture, and then sort of the complexities of eukaryotes. So we already sort of talked about this. When I gave that example, we talked about how bacteria, because it's like a one-cell entity, it has to respond very quickly to its surroundings. If there's nutrients that are gone, it has to quickly switch which nutrients it uses. If there's a heat shock, it's got to change the genes. If it's, invading a host, it's got to change its gene expression pattern sometimes. So they're going to quickly be responding to environmental changes. This right here is a scanning electron micrograph of your gut bacteria. And I always like to just share this fact that we actually have 10 times more bacterial cells in our body, in our different floras, than we do our own human cells. So it's kind of amazing that we're actually hosting all these things that are so tiny, more cells than we actually have. But the gut bacteria, they're responding to constant changes in your diet to figure out, what are they using as nutrients. So if you switch what you eat entirely, that can really affect your gut bacteria, right? It can affect their gene expression for them. So today we'll talk about how do bacteria sense the nutrients that they have available, and then how would they switch in the case, let's say you go on some crazy low carb diet and you're going to take all glucose out of your diet. How would a bacteria respond to that? So if we look at this, come back to this, we talked about now replication, transcription, and translation. If you were going to want to change the proteins that are available in your cell, which places could you regulate? Which processes could be regulated? Yeah,
Which transcription factors are turned on?
yeah. So you could affect transcription step by maybe regulating like what genes are being turned on. Yeah. Any other places could you also regulate translation? Could. There's ways to regulate the ribosome. With the protein, like let's say that you wanted to get rid of a protein, are there ways to degrade proteins? So really you could regulate it at the point of transcription, translation, or even post-translation. So these are the different places that you could regulate. Which of these do you think is gonna be smarter so that you don't waste energy? Yeah, so why do we say that? That is the best place to regulate if you don't wanna waste energy. Why would that be? Yeah, Nat?
Early on, so you're not doing all the stuff that you're not gonna do.
Yeah, exactly. So you're gonna, before you go to all this effort and use all the ATP for the charging and all of the stuff that you have to do to translate, you stop it early. Which place do you think would be the fastest if you just needed to quickly get rid of the protein? Which step do you think would be the best place to regulate? Yeah, so that's gonna just get rid of the protein, and it doesn't take all this like powering down. Now we're powering down transcription. We still have a few mRNAs that are already in the pipeline. Right, so this is the fastest, but also the most wasteful. Transcription is slower to do, but it's more energetically favorable place to regulate. Okay, so this one would be energetically expensive, but also the fastest. Okay, so we are going to get into, for we're talking about gene regulation, there's a lot of new terms. So I wanted to kind of go through some terms so that you have kind of a record, or we start with that framework, and then we can kind of, as we go through and talk about different proteins, and we talk about the LAC-OPERON, you can kind of be classifying things as trans-acting elements or cis-acting elements. Okay, so we've talked about the DNA of a gene. In this case, this is showing something in a eukaryotic cell, because this is taken from a textbook. But these terms are going to apply to both eukaryotes and prokaryotes. Here's where the gene is, and then this is showing, here's the core promoter. This would probably be the proximal promoter. The trans-acting elements are going to be anything that, I just remember trans is like moving across. So these are things that move across the cell to regulate at these sites. So trans-acting elements are typically going to be proteins. In all the systems that we talk about, we're talking about trans-acting elements. They're usually the regulatory proteins. And for your notes, you can include some examples. They could be like transcription factors. They don't have to be always activating. They sometimes are repressing. So they can be activators or they could be repressors, but they're typically proteins. Cis means next to. So cis-acting elements are things that are right next to the gene that regulate that gene. So cis-acting elements would be DNA elements like promoters or enhancers or other pieces of DNA that control the synthesis of the gene. Okay, so here your promoters would be cis-acting, all of these would be trans-acting. All right, and then kind of some further terms, just make sure we have all of these or we've heard of all of these before. Activators are generally thought to be positive regulators. That means they increase transcription. Repressors are going to be those proteins that are negative regulators that are decreasing transcription. And then for cis-acting elements, again, they're mostly DNA sequences. And so some examples for the LAC-OPERON, the promoter sequence is a cis-acting sequence. The operator is actually going to be found next to the promoter, but it's also a DNA sequence, so it's considered cis-acting. And then for the LAC-OPERON, there's another piece that you don't always learn about in Biology 160. It's called the activator binding site. And so that's also a piece of DNA. So it's going to be cis-acting. Okay. So two other terms. Well, first of all, anybody have questions on those terms? Okay. These are the last two terms that we have to kind of before we talk about the LAC-OPERON. The first is constitutive. I just usually remember constant sounds like constitutive. So those mean the same thing. Constitutive genes are genes that are always turned off, for the most part, constantly turned on. So these would be like housekeeping genes, genes that you just need for everyday cell life. So I want you to think for a second, what types of genes would be always on. You don't have to have names of genes, but just general classes of genes. Can anybody think of something that you're just always going to need in the cell? The cell always has to do. Yeah. Yes, exactly. So you always have to make energy, right? You always have to be able to produce ATP. So you could put, for an example, the genes that are involved in cell respiration. So that's kind of what you're thinking of. Those, you know, they could be involved in like glycolysis or any of those other processes that you need to produce ATP. The other type is inducible. So some genes, you don't need them unless there's some kind of situational need for that gene. So the example of that would be in the lac operon system. You don't always need to make lactase, or not lactase, beta-galactosidase. You don't need that enzyme unless you have lactose there that you're going to be using. So that would be situational, and you don't want to waste it because actually the cell is going to prefer to use glucose if at all possible. So that would be an example of an inducible gene. So those are kind of our basic gene regulation terminology. So for the lac operon, sometimes people say, why do you always teach, why do we still teach about this? This system was discovered in the 1970s. And it's actually phenomenal what they did at that time because they didn't have stuff that we have today. They did not have DNA sequencing. They didn't have a lot of the molecular techniques that we have. And they figured this all out like a giant puzzle using genetics. So this entire system was figured out using genetics. And that's actually where those terms like cis-acting, trans-acting, they came from these scientists, Jacob, Monod, and Bloch. In their sort of system, they kind of figured out what factors had to be acting across and which factors had to be right next to the genes. And so to me, it's actually phenomenal that they were able to do it just by figuring out the puzzle, just by using genetics. They were also the ones that coined the term operon. So we already talked about operon. Does anybody want to just remind us what an operon is? This was in our gene expression lecture. Yeah, that one.
Is operon where just one set of regulatory sequences can turn on multiple expression proteins?
Yes, exactly. So that is very common motif that we find in bacteria where they have one set of regulatory sequences is like one set of light switches that turn on a whole bunch of genes at once, right? Okay, so let's talk about their system here. This is the lac operon system. In general for cells, the preferred energy source, what is the preferred sugar to provide the energy? Yes, so this blank should be glucose. So if glucose is ever present in the cell, that's what the cell is going to use. It's pretty much all the metabolic pathways are optimized for using glucose. But if you don't have it, you still need to have some kind of energy, so you can use alternative sugars. So these are a couple of alternative sugars that bacteria can use. So lactose is milk sugar. Maltose is found in like barley, or if you break down starch, you produce maltose. And then arabinose is another alternative sugar that bacteria sometimes use. They do not, because they're not always using these, they basically don't turn on the enzymes that are needed to break down the sugar unless they're using it. So all of these, lactose, maltose, arabinose, they all have their own operons. And you would induce those operons if there's no glucose, but you have that other sugar. Okay, so we're going to kind of look at lactose as the model, but you can kind of apply all of what we learned about lactose to all these other sugars as well. Okay, so the pieces that we need for breaking down lactose, there are basically 3 different genes that are in the lac operon. Let me kind of draw this out, and I think this is part of your first question on the ICA here. So we'll start by putting the genes in there. There's 3 genes, LAC-Z, LAC-Y, and lact-A. Okay, and each of these produce a different protein that's needed for some part of this process. Lact-Z, you can see, produces this enzyme right here, beta-galactosidase. Sometimes people just call it beta-gal. I'll just write beta-gal. You can write down beta-galactosidase. That is produced by lact-Z. This is probably the most important enzyme because it's actually the enzyme that catalyzes the cleavage of lactose into galactose and glucose. So this is our disaccharide, and then it cleaves it into two monosaccharides. Okay, the second enzyme, enzyme that is produced is lact-Y. And lact-Y produces the permease. So you see this big purple permease here? This essentially lets the floodgates open to just let lots of lactose into the cell. So if you decide this is the route we're going to go, we're going to use lactose, you need to let as much lactose into the cell as possible. So this allows lactose in to the cell. The third enzyme is not pictured here, and actually a lot of textbooks don't really even talk a whole lot about it. This is a transacetylase, and it is used to chemically modify lactose. We're kind of, for the most part, going to leave this part out for now, but just know that it does produce this enzyme that can chemically modify lactose. Okay, so those are the three parts. We have them here. Lact-Z, lact-Y, lact-A. Those are called structural genes because that's actually the products. That's like what you're trying to turn on. The regulatory sequences are to the 5' end, and they include, so in your picture you want to draw these on next to the genes. The first thing is the operator. Now the operator is positioned there for a reason. If you're going to shut this whole thing down, you have to have kind of a way to lock it down. So repressor protein can bind at the operator and block the RNA polymerase from binding at the promoter. So that's why the operator is kind of standing in between the promoter and the actual genes. So here's your operator. This one binds to a protein called the repressor. The lac-I repressor. So that will be a protein. If it physically binds right here, you can see that that RNA polymerase wouldn't be able to bind at the promoter. Okay, so here next to it is the promoter. And then I actually want you to change the picture a little bit. This one comes from one textbook. There is another thing to the right of the promoter, which is called the activator binding site. So let me label that here. Activator binding site. And this is going to bind to a protein called CRP. So binds to CRP, which is going to be an activator.
Is that what they test for when they do like a CRP test and blood test?
Oh no, that's a C-reactive
Oh.
protein. But yeah, same initials but different protein. Okay, so we added that right next to the promoter for the lac opera. And then on your picture, you can add these kind of like hash marks to say like upstream, further away, you have, now I'm running out of space, you have lac-I gene with its own promoter. The lac-I, what did I say lac-I was? It's a repressor. So you have to make the repressor from a different location. The repressor as a protein is going to float over and bind right here. Okay, so does everybody understand all the pieces here? So for a second, I want you to talk to the person next to you. Which of these is cis-acting and which of these is trans-acting elements. And you can circle your cis-acting elements on there. You might have to add in some of the things that you know are supposed to bind to those sequences to kind of label all the cis-acting elements.
Oh, this is what I see.
Trans increases transcription.
So, wait, trans is the one that looks,
No, those are accurate. Trans always find regulatory expressions for sequences and the other vectors.
so,
So black
black
eye
eye
would be
would
trans.
be, yeah, trans.
And then
Cis would be,
okay, got it. Okay, so yeah, the long, the
yeah,
protein is
okay,
considered a
but I
trans.
wasn't sure.
I could type it over there. I don't know why.
It's exacting because it's next to the G.
And also one other note is that the structural genes, you don't really count them as cis or transacting because they're not regulatory elements. They're just the products.
Okay.
So you don't have to classify Z, Y, and A as anything.
Let's see, the activated binding site, that itself is cis, but it binds to the activator.
So promoter would be
That is.
definitely elusive, right?
Switch seats, we can switch seats. There's another seat here too. Okay, and the lac I, that itself is a, it creates the repressor. So the repressor creates is trans, but it itself is cis.
That's a little bit like,
This is we're talking about gender again. It's gonna be a weird one to wrap my head around. So lac I is protein trans. Yes, the lac I, no, the lac I encodes the repressive protein, but then there's the lac I repressor itself.
Operator.
well, I think like I
But the
should be
operators is what?
Oh, yeah.
like I cap is all
Cis.
promoted instead of regulate.
Oh, okay, so you don't have to talk about that.
Is it
So,
just
these, okay,
Do you have to do
then
these
try to
two?
draw this. I think I know, I want to erase it and draw it.
Doesn't matter.
I'm so glad I did it on my own first.
I know the structural ones you said are just. they're
Okay, let's
just
see what you all
structural.
are thinking about for which ones that we have here. It's kind of like I'll show them over here. Which of these are, let's just start with the trans-acting, or cis-acting, circle the cis-acting elements. Which things did you circle, or do you think you should circle? So you wanna,
Faster,
what'd you say?
faster.
The repressor, okay, so that's a question. Is that one, the repressor, is it cis-acting or trans-acting?
Oh, transacting.
Okay, yeah, so repressor, we're gonna say that that is trans-acting. So this lac I repressor, as a protein or as the DNA sequence, this sequence is also this is called trans-acting because it produces the protein that is then gonna move and regulate. So circle, oh, so that's not cis-acting, square, right? I think I put a square around it. Square around the protein. That is trans-acting because it floats through the cell and it binds to the operator sequence. Any others that were, I guess you can say whichever factor, and you can say if it's cis-acting or trans-acting. How about this one, operator, is that cis or trans?
Cis.
Cis, right? So circle, circle the cis, yes. This is a cis-acting element. How about the promoter?
Cis.
Cis-acting. How about the promoter for the lac I? Cis. So it's interesting because because the promoter is cis-acting, but the protein that is encoded is transacting. The other things we didn't actually, I didn't draw here, but the CRP protein, what would that be? Trans. So I'm going to put a box around that because that is a protein that binds. Now here's the question you can always ask yourself to test out if something is cis or trans. If you move it, you move that element to some other location in the genome, would it still work? If it has to be next to the thing that it's regulating, then it's a cis-acting element. For example, if I took this operator and I moved it way far upstream, could it still block the promoter from that new location? No. So that tells me it has to be cis next to the thing that it's regulating. If I moved this repressor though, what if I took this gene and I moved it somewhere else? Would it still do its job? As long as the protein is being made? Yeah, so that tells me that this is transacting. I can move it somewhere else. Its location doesn't matter as long as it's being produced. So that tells me it's a transacting factor. Okay, so I think we got all of number 1 done now. Does anybody have questions? Yeah.
Is the activator binding site itself, not the CRP, is that considered a cis?
Yeah. So, sorry, I never, oh, circle, sorry. My own notation, yes. The CRP binding site, or you say activator binding site, that is cis. Cis as well. Okay, so let's look at how this works now. So again, because I told you LAC-A, sometimes textbooks sort of ignore it. It's just not even shown here in this. This is from our textbook. But this now shows you the position of these genes, what they make, and also the LAC-I, and what that makes. So the question is, when would I want to turn this whole system on? When would I want to make LAC-Z and LAC-Y? What would be the conditions?
Only when there's lactose?
Only when there's lactose, and what would have to be true of the glucose situation?
No.
No glucose. So even if, let's say I have lactose, but I also have glucose, I still don't want to use this because the cell prefers to use glucose. So let's look at, all right, what if we don't have lactose around? What happens in the absence of lactose? What happens it's sort of a default system, and that is because this LAC-I gene produces this repressor in a constitutive manner. I'm going to write that down. This is constitutively on, meaning that the repressor is always being made. So I'm always, by default, producing a whole bunch of repressor protein. If there's no lactose in the cell, that repressor is made and it's binding at the operator. So if you want to annotate on here, you could label this the operator. This is where the promoter is. So by default, that binds, and then it basically, as you can see here, will block RNA polymerase. So would I produce LAC-Z, LAC-Y in this situation? No, not at all. I wouldn't even get any transcription. So this is off. And the repressor is very tight. It keeps it completely off. Okay, let's zoom in a little bit more about this interaction right here. So we're going to look at what is the operator sequence and what is this weird protein. It looks like to me that looks like when you blow up a glove, you know. And I'll explain why they drew it like that. So the operator sequence is actually, as you can see here, if you read this sequence and this sequence, it's the same, right? So that's called an inverted repeat, which means that two LAC proteins can bind. So it binds as a dimer, and the LAC operon is actually a tetramer, so there's even another position where the LAC repressor can bind at another site. But here you can imagine here's one piece of one LAC protein, and the other LAC protein is binding over here. So that each of those proteins binds with one of the half sites to shut this down. And how does this prevent binding? Well, if you look at where this operator is, Now it kind of shows you the context with the +1 site. So what does +1 mean again? We already talked about this with transcription.
That's where the first nucleotide binds.
Yeah, this is where you first start transcribing at the +1 site. The operator is actually kind of inside of that. So if there's a protein mapped to this position, it overlaps with the position where the RNA polymerase would bind. So this physically actually won't even let RNA polymerase bind if there's a repressor there. This picture that is from your textbook is inaccurate because it's showing them binding at the same time. They actually physically overlap. So if the repressor is there, there will not be any binding of the RNA polymerase. So for this blank, we can say repressor binding at the operator prevents or blocks RNA polymerase from binding at the promoter. Okay, just to kind of give you the relative position, I've pointed out now and I'll come back to it. This is the activator binding site. It's really close to where the DNA site where RNA polymerase binds, and that's going to come into play because this cap is the same as CRP. Anyway, in science there's always multiple names for everything, but the protein, when it binds here, it can actually help RNA polymerase to find that site easier. So I'll come back to that. I just want to show you when they're all shown next to each other so you can kind of understand that later. So this first level of regulation by this repressor, this is considered negative regulation. So hopefully you can see that the repressor prevents transcription. That's negative. So it's decreasing transcription. And that is all for the lactose sensing mechanism. Okay, now let's talk about what if we have some lactose in the cell. So now we actually might want to use lactose. So what happens when lactose is there in the cell? Lactose is called the inducer because it's going to turn itself on. It's going to turn its own operon on. It physically binds to this repressor protein and changes the conformation. So when the protein changes conformation, it can no longer bind to the operator sequence. So all of a sudden it pops off. And now it leaves it wide open for the RNA polymerase to be making LAC-Z and LAC-Y and LAC-A. Okay? But we said that we don't necessarily want to use this. What would be a situation where we would want to transcribe? If lactose is there and what else has to be true? No glucose. So the next question is, how do we then sense for glucose? Because we don't just want to let it transcribe necessarily. And that brings into play the CAP site, or the activator binding site, whatever you call it. So here, this is kind of walking you through this. If glucose is high, but lactose is also high, if the lactose is there, it's going to get the repressor to fall off. You can get a little bit of transcription. Now, I wrote here, or the textbook wrote, infrequent transcription. We can call that basal transcription, like low levels. Not basal, the herb. I had somebody on the test write like basal transcription. But I mean basal, like low levels. That's like a little bit of transcription. There's going to be some leaky transcription just because I'm not blocking this off, so you can get a little bit. But the issue with this is that the promoter is not a good promoter. So do you remember when I showed you the -10 and the -35 sequences, and I said if it matches perfectly, you get really good transcription, but if you have mutations away from that sequence, it's not a strong promoter. So LAC does not have a strong promoter, and that's why you only get infrequent transcription. But you would get some. If, however, you have low glucose, now you see what happened. This little squiggly thing came in. So that is representing the CAP or the CRP protein is coming in. It's binding at that purple site, which is the activator binding site. And that helps to pull RNA polymerase to the correct position. So it actually helps to improve transcription because it's recruiting RNA polymerase to come to the right site, even though there's a very bad promoter there. So only in this case would you get plus 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 transcription levels. That means we get lots of transcription, okay? Only when glucose is there, or yeah, glucose is not there, sorry, and lactose is there. Okay, so let's see if we can answer these questions. Even in the presence of lactose, the lact promoter is weak. How could it be weak? Or how do you explain that it's a weak promoter?
It doesn't conform to the standard promoter strength.
Yes, exactly. So that's how you can answer that question, is that the -10 and the -35 sequences don't match exactly. So it's not a strong promoter. Okay, so we get a little bit of transcription in this case. The lac operon is only turned on when the lactose is high and glucose is low. And the reason for that is that when glucose is low, that's the only time that the cap protein will bind at this activator binding site. Okay, and I'm going to explain why that is in a couple of slides. So, the cap part is positive regulation. We just talked about how the repressor piece was negative because it decreased transcription. But by having this cap protein, that is increasing transcription. So this is called a positive regulator, and this is the negative regulator. Okay, so the cap also is the thing that is able to sense the glucose levels. Okay, so let me just summarize here. We already said that sensing of the lactose levels is done by the repressor, and that is a form of negative regulation. In the absence of lactose, we're going to have a repressor bound. It's going to block here, and we'll get no transcription. So that would be the scenario of this, no transcription. And then positive regulation, let's say that lactose is present and glucose is not there. We will then have this positive regulation where the cap protein will bind to that cap site, and it will help to recruit the RNA polymerase. So are these pieces kind of coming together? Yeah.
Is the cap produced at a different part of the operon that is only produced when there is a low presence of glucose?
Oh, so I will explain how it will know when the glucose levels are there. Yeah, that's a good question. So this whole system, it has to be able to sense glucose. And the way that it does this is not the best slide to explain that. How does CAP sense glucose levels? Well, CAP has to bind and can only bind when it has cyclic AMP. Where did we learn about cyclic AMP before? It was another lecture. Signaling, right? We talked about cyclic AMP as a second messenger, the small molecule, but it turns out cyclic AMP can bind to this CAP protein, and only when it is bound, that's when CAP can bind to the actual activator binding site. So the levels of cyclic AMP are sort of a readout for whether glucose is there or not there in the cell. And this is the relationship, and then I'll explain to you why this relationship exists. In high glucose levels in the cell, there will be low cyclic AMP. If there's low glucose, then there's going to be high cyclic AMP, and that's when cyclic AMP can bind to CAP, and they can bind to the activator binding domain, or binding site, sorry. So, this is, it just as a reminder, we actually already looked at this exact same slide, showing you how you produce. This right here is ATP, adenylate cyclase, cyclizes, remember, and this produces cyclic AMP. Glucose transport reduces the level of cyclic AMP by inhibiting this enzyme. So when you have high glucose, you're going to have low cyclic AMP. So that explains the relationship. For me, I don't care if you just can remember this relationship right here. High glucose is low cyclic AMP, or low glucose is high cyclic AMP. If you remember that that's inversely related, but sometimes people like the explanation, why is that? And so this helps to explain why that is. All right, so does anyone have questions so far? Okay, now I think we can do this part. We're gonna do this, I think we'll kind of divide it up and then that way we can go over in class. That'll be maybe the last thing we have time for. So, how about you can do A, and this group of three can do B, and then maybe the three of you can do C, and you all four of you could do D. So what you are doing for this is actually drawing out, just like we drew out this. So you want to draw the same picture that we drew, and then you show the pictures of whether the repressor is there, whether the RNA polymerase is binding, or the cap protein. And you have to decide, are we getting minus, plus, or plus, plus, plus for transcription? Go ahead and talk, you can go through the one that you're working on.
Wait, so yeah, there's nothing, there's gonna be minus here. But does it have everything?
I think it's gonna be the negative.
Oh, no, yeah, we could do it together. Yeah, we can work it together.
Okay, so, now.
You should bring a chair. Yeah, absolutely.
I think it's the knot because there's
It's gotta be the knot.
Oh, I'm gonna do that.
I wouldn't like it expressed. There's no like this, so it'll be fully inhibited, right? Like the beginning.
No glucose. I
Yeah, because the camp would find the G and then inhibit, yeah, lactose, but then there's none on those. So, I don't know, is there even anything at this? I don't.
Yeah, so there'd be no lactose. So
They have a
repressor
nicer.
would be activated, which covers up the